Good day, this is Professor Will, CBMD PhD. Today's Sunday, August 25th, 2019. It is 18:04:35 seconds Eastern Standard Time. And we are now going into DNA restriction modification on my research for pathophysiology. So let's hop to it here. Awareness of the biological phenomenon of restriction and modification (RM) grew from the observations of microbiologists that the host range of bacterial virus phage was influenced by the bacterial strain in which the phage was last propagated. Although phages produced in one strain of Escherichia coli would readily infect the culture of the same strain, they might only rarely achieve the successful infection of cells from a different strain of E. coli. This finding implied that the phages carried an imprint that identified their immediate provenance. Simple biological tests showed that the occasional successful infection of a different strain resulted in the production of phages that had lost their previous imprint and had acquired a new one. In the 1960s, elegant molecular experiments showed that imprint to be a DNA modification that we lost when the phage DNA replicated within a different bacterial strain. Those phages that conserved one of their original DNA strands retained the imprint or modification, whereas phages containing two strands of newly synthesized DNA did not. The modification was shown to provide protection against an endonuclease, the barrier that prevented the replication of incoming phage genomes. Later it was proven that the modification and restriction enzymes both recognize the same target and specific nucleotide sequence. The modification enzyme was a DNA methyltransferase that methylated specific bases within the target sequence, and in the absence of specific methylation, the target sequence rendered the DNA sensitive to the restriction enzyme. When DNA lacking the appropriate modification imprint enters a restriction proficient cell, it is recognized as a foreign and degraded by the endonuclease. The host control barrier to successful infection by phages that lack the correct modification was referred to as a restriction, and the relevant endonuclease have acquired the colloquial name of restriction enzymes. Similarly, the methyltransferase are more commonly termed modification enzymes. Classically, a restriction enzyme is accompanied by its cognate modification enzyme, and the two comprise the RM system. Most restriction systems conform to this classical pattern. There are, however, some restriction endonucleases that attack DNA only when their target sequence is modified. A restriction system that responds to its target sequence only when it is identified by modified bases does not, therefore coexist with a cognate modification enzyme. An understanding of the restriction phenomenon observed by Luia and human requires knowledge of the special nature of the DNA of T-even phages. During replication of T-even phases, the unusual base 5-hydroxylmethylcytosine HMC completely substitutes for cytosine in the T phase DNA, and the hydroxyl methyl group subsequently glycosylated in the phase-specific pattern at the polynucleotide level. In the mutant strain of E. coli used by Luia and human as host for the T-even phases, glycosylation fails, and in its absence, the non-glycosylated phase DNA becomes sensitive to an endonuclease present in E. coli K12 but not in the Centuria. Particular nucleotide sequences, normally protected by glucosylation, are recognized by an endonuclease in E. coli K12 when they include the modified base. HMC rather than cytosine resides. In the T-phase experiments, the modified base is hydroxylcytosine, but much later it was discovered that the methylated cytosine resides can also evoke restriction by the same endonucleases. The classical RM systems and the modification-dependent restriction enzymes share the potential to attack DNA derived from different strains and thereby restrict DNA transfer. They differ in that, in one case, an associated modification enzyme is required to protect DNA from attack by the cognate restriction enzyme, and the other modification enzyme specified by different strains impart signals that provoke the degradative activity of restriction endonucleases. Phages often provide a useful and sensitive test for the presence of RM systems in laboratory strains of bacteria, but they are not a suitable vehicle for the general detection of barriers to gene transfer. Many bacterial strains, even within the same species, and particularly when isolated from natural habitats, are unable to support the propagation of the available test phages, and some phages, e.g. P1, have the means to antagonize at least some restriction systems. Gene transfer by conjugation can monitor restriction, although some natural plasmids, but probably not the F factor of E. coli, are equipped with anti-restriction systems. The single strand of DNA that enters a recipient cell by conjugation or following infection by a phage such as M13 becomes sensitive to restriction only after the synthesis of its complementary second strand, whereas a single strand of DNA that transforms naturally competent bacteria may not become a target for restriction because it forms heteroduplex DNA with resonant and therefore modified DNA, and one modified strand is sufficient to endow protection. Transformation can be used to detect restriction systems, but only when the target DNA is the double-stranded DNA of a plasmid. In vitro assays for DNA fragmentation, endonuclease activities yield discrete fragments of DNA are commonly detected in crude extracts of bacterial cells. More than one DNA may be used to increase the chance of providing a substrate that include target sequences. DNA fragments diagnostic of endonuclease activity are separated according to their size by electrophoresis through a matrix, usually an agarose gel, and are visualized by the use of fluorescent dye, ethidium bromide, that intercalates between stacked base pairs. Extensive screening of many bacteria, often obscure species for which there is no genetic test, has produced a wealth of endonucleases with different target sequence specificities. These endonucleases are referred to as a restriction enzyme even in the absence of biological experiments to indicate their role as a barrier to the transfer of DNA. Many of these enzymes are among the commercially available endonucleases that serve molecular biologists in the analysis of DNA. In vitro screens are applicable to all organisms, but to date restriction and modification systems have not been found in eukaryotes, although some algal viruses encode them. Sequence-specific screens. The identification of new RM genes via sequence similarities is sometimes possible. Only occasionally are gene sequences sufficiently conserved that the presence of related systems can be detected by probing southern transfers of bacterial DNA. Generally, screening databases of predicted polypeptide sequences for relevant motifs has identified putative RM systems in a rapidly growing list of bacteria for which the genomic sequence is available. Currently, this approach is more dependable for modification methyltransferase than for restriction endonucleases, but the genes encoding the modification and restriction enzymes are usually adjacent. Many putative RM systems have been identified in bacterial genomic sequences. Nomenclature and classification. RM systems are designated by the three-letter acronym derived from the name of the organism in which they occur. The first letter is derived from the genus, and the second and third letters from the species. The strain designation, if any, follows the acronym. Different systems in the same organism are distinguished by Roman numerals. Thus, HIN2 and HIN3 are two enzymes from the Haemophilus influenza strain RD. Restriction endonuclease and modification methyltransferase, enases and mtases are sometimes distinguished by the prefix R eco RI and M eco RI, but the prefix is commonly omitted if the context is unambiguous. Classification of RM systems. RM systems are classified according to the composition and cofactor requirements of the enzymes, the nature of the target sequence, and the position of the site of DNA cleavage with respect to the target sequence. Currently, three distinct, well-characterized types of classical RM systems are known, 1 and 3, although a few do not share all the characteristics of any of these three types. I should conclude this reading with the glossary. ATP and ATP hydrolysis. Adenosine triphosphate, ATP, is a primary repository of energy that is released for other catalytic activities when ATP is hydrolyzed, split to yield adenosine diphosphate. Bacteriophages, lambda and T-even bacterial viruses. DNA methyltransferase, enzymes mtases that catalyze the transfer of methyl group from the donor S-adenoacyl-methylamine to adenine or cytosine residues in the DNA. Efficiency of plating. This usually refers to the ratio of the plaque count on a test strain relative to that obtained on a standard or reference strain. Endonucleases, enzymes that can fragment polynucleotides by the hydrolysis of internal phosphodiester bond. Escherichia coli strain K12, the strain used by Letterberg and Tantum in their discovery of recombination in E. coli. Glycosylation of DNA. The DNA of T-even phages, in addition to the pentose sugar deoxyribotes, contains glucose attached to the hydroxyl methyl group of hydroxylmethylcytosine. Glucosylation of the DNA is mediated by phage-encoded enzymes, but the host provides the glucose donor. Helicases, enzymes that separate paired strands of polynucleotides. Recombination pathway, the process by which new combinations of DNA sequences are generated. The general recombination process relies on enzymes that use DNA sequence homology for the recognition of the recombining partner. SOS response. DNA damage induces expression of a set of genes, the SOS genes involved in the repair of DNA damage. Southern transfer. The transfer of denatured DNA from a gel to a solid matrix, such as a nucleocellulose filter, within which the denatured DNA can be maintained and hybridized to labeled probes. single-stranded DNA or RNA molecules. Fragments previously separated by electrophoresis through a gel may be identified by hybridization to a specific probe. Transformation, the direct assimilation of DNA by a cell, which results in the recipient being changed genetically. Good day, this is Professor Will, CBMD, PhD. Today's Sunday, August 25th, 2019. It is 19:52 and 47 seconds Eastern Standard Time, and this concludes my reading on DNA modification and restrictions. Hope you enjoyed listening. Have a great day.